magnification. You're asking about to calculate magnification based on the scale bars. How do you solve this exercise?
First, I measure the line, the black line, like you see, and it was like 22 centimeters. I converted into millimeters. Then
Uh-huh.
I divided the 20 millimeters to the 67. I don't know how to say that, like
Micrometer.
micrometers, and I don't know. Sorry, the 20 millimeters I converted into micrometers. Then I divide
Mhm.
those micrometers with the 67, and my answer was 298.5 times.
Okay. 298 times. That's the magnification of this scale bar. Okay. Any questions, guys, about how to solve this exercise?
Teacher,
So-
it's
yeah.
not like about how to solve, but like giving out the answer. So like if a magnification ends in a decimal point, do we need to write it down,
Yeah, you need to.
or
No, you
we
need to
just
run the. You need to round
so
off
if it is
the
like
figure, guys.
okay, okay, thank you.
Yeah, since we're talking about the magnification, it's recommended to round the figure. Okay. The second exercise, you were given a picture, an electron micrograph of cells, and you're also given the magnification. And they ask you about to calculate the actual size of the cell. What do we have to do, guys, in this exercise?
Measure
Uh-huh.
the diameter, like that's kind of like in the yellow line, and
Uh-huh.
then divide that by the magnification, and then convert it to, I don't know what this measurement is. I don't think it's nanometers, but
Okay. So we measure the cell, and since this image has been magnified 1,000 times, so it's 1,000 times smaller, right? So we divide this measurement by the 1,000 times. Okay. Probably you measured the cell in centimeters, right? Or millimeters.
yeah.
Yes.
Yes.
Whenever we are asked, guys, about to measure and calculate the actual size of a cell, we we must have micrometers as a unit. Okay, since what's the size of a cell? Prokaryote cells are between 1 to 10 micrometers, and eukaryote cell between 10 to over 300 micrometers. So the recommendation is to use micrometers as a unit, okay, for the measurements for cells, or nanometers, like in this exercise, micrometers and nanometers. If, for example, we're measuring an organelle or DNA as a molecule, then the recommended unit is, since we're talking about microscopic objects, we need to use micrometers as a unit to express their size. Okay. So if you measure this image in millimeters, it's about 40 millimeters. 1 millimeter has 1,000 micrometers, so we have 40,000 over the 1,000 times of the magnified. We get the size about 40 micrometers, right? Any questions, guys?
No.
Okay, what about exercise number
Teachers,
3?
so in that,
Sorry?
I'm sorry, I had like the doubt at the final, at the end, sorry. And so in that case of the second problem, we always need to write it on in micrometers by later by the length, sorry, by the magnification. Like we need to calculate the length of the line and we need to convert it into micrometers, right? To do
Into
all the procedure.
micrometers. Yeah. Since again,
Okay.
we're measuring cells, and cells, their size is expressed by their size is expressed in micrometers. That's the recommendation, guys. Because otherwise, you can use, I don't know, millimeters, centimeters, even meters, but the recommendation is to use the units, or uh, someone else asked me about the number of significant figures. You can either use the decimals or you can use either in the scientific notation. It's the same, but you can tell that the units are the same, micrometers. That's the recommendation, guys. Okay.
Thank you, teacher.
How about exercise number 3? Yeah.
What I did was firstly measure the, we can say the X line. Like I did a
Mm-hmm.
simple rule of three, three, and I said like the
Mm-hmm.
20 ohm line measures 1.65 centimeters, and if the other one measures 6.8, what would be the like the quantity of ohms? And it gave me
Okay.
out like 82.44. And
Mm-hmm.
after that, I calculated the magnification. What I did was the 6.8 centimeters converted into micrometers that gave me out 68,000 and divided into the length, the 82.44, and it gave me a magnification of 825.
Mm-hmm.
And after having the magnification, I needed to calculate the length. No, sorry, the actual size. And what I did was the again the 68. Sorry, but I think that here I have the error because I did it with millimeters.
Okay.
So, but the final
Yeah,
step
there,
is
there.
dividing the 68,000 ohm between the magnification.
Okay. Yeah, you have two options here, guys. You can either calculate the magnification, and then based on the magnification, you can calculate the actual size, or as you said in the beginning, you can measure this line. Okay, and the question is how many this scale bar can fit within this line, and that number of times the value of the scale bar, and we get the actual size of the cells. So you have two options. Just as long as you get the correct answer and you show you're working, it's okay. And the same goes for the rest of the exercises. You're given the length of the cell, the scale bar with the value. In this exercise, you're not given the line, but you can measure. It says the diameter of the main body of the human cell. Here the same. You need to calculate the length. We measure the length of the cell. We divide this value by the size of the scale bar times its value in order to calculate the length of this cell. So, any questions, guys, about to calculate the magnification or the actual size of an object?
No.
not really. Yeah, these are quite easy exercises. Okay, if there are no questions, guys, then we're done with this topic. Okay, topic 1.2. And let's continue with the next one, that is 1.3, the membrane structure, and also 1.4, membrane transport. So these two topics are focused on the structure of the plasma membrane of the cell and the way the cell uses transport mechanisms to move substances across the plasma membrane. So 1.3, the structure of the membrane. The essential idea for this topic is that the structure of biological membranes make them fluid and dynamic. Here are the understandings, applications, and skills. And the second topic, that is about transport mechanisms. The essential idea is that membranes control the composition of cells by either active and passive transport. So these topics are about the structure of the membrane and the way the membrane controls and regulates by several mechanisms the movement of molecules across the structure of the membrane. Okay, let's start by defining the function of the plasma membrane. First of all, it forms a boundary layer that separates what's inside of the cell from the surrounding environment of the cell, so that the cell can control and regulate its composition by means of the plasma membrane. All the materials that are needed or produced by the cell can remain on either side of the membrane. Again, it will depend on several factors. That was the main role of the plasma membrane, is to control and regulate its composition. So this structure is about 8 nanometers thick. Okay, and it says that it has a selective control of the movement of materials into and out of the cell. This has selective control is based on several factors. Okay, it depends on the size of the molecule, whether it can be moved or not across the membrane, or the type of mechanism used by the cell to move this molecule. Another factor that must be considered is the chemical nature of molecules. Since we're talking about an inner and an outer environment that are composed of mainly water, we're talking about hydrophilic environments. And sometimes, substances can be hydrophilic, some other times can be hydrophobic, but also the structure of the plasma membrane, due to the composition of the plasma membrane, is how it is selective and controls what kind of molecules based on their chemical nature, hydrophilic or hydrophobic, and the type of mechanism used, either active or passive transport, to move the molecule across the membrane. Okay. Another factor that must be considered is the composition of the external and the internal environments based on the needs of the cell. It depends on the concentration of the molecules the way these are going to be moved to either side, and also the type of mechanism used by the cell. So their size, the chemical nature, and the composition of both the inner and the outer environment play an important role in the selective control of the movement of materials across the membrane. Another function of the membrane is that it's responsible for the recognition between cells. It plays an important role in the way cells that can be considered alike, that might have the same function, can be recognized to each other. The way cells recognize as a part of an identical type of cell with an identical function, so that they can be organized into a higher level into tissues. This is the way tissues are formed, and then these tissues can be further organized into a higher level as organs. And this is due to the structure and composition of the plasma membrane. That is actually another feature. It says that the structure of the plasma membrane is dynamic, meaning that it has a distinctive inside and outside composition. What's in the inner face of the membrane can be different from what is in the outer face of the membrane. So the inner and the outer faces of the cell membrane can be different, and they can change according to the needs of the cell. Okay? Now, the first attempt to describe the structure and the composition of the plasma membrane, it was by Davson-Danieli. They proposed the structure of the plasma membrane based on a phospholipid bilayer. Okay, we already talked about these molecules, phospholipids, that are described as amphipathic molecules, meaning that they have a hydrophilic portion and a hydrophobic portion within their structure. They can have both behaviors, hydrophilic and hydrophobic, meaning that they can interact, form hydrogen bonds with water, or they can repel water. Just to recall that phospholipids are composed of an alcohol, glycerol, 2 fatty acids, that could be either saturated or unsaturated, and a phosphate group. So the hydrophilic portion consists of the glycerol, and the phosphate group. This is going to be referred as the hydrophilic head. And another portion, the one that is said to be hydrophobic, also known as the hydrophobic tails, are the 2 fatty acids. So one portion of the phospholipid is attracted to water, another portion is repelled by water. And these phospholipids are organized in this configuration within the phospholipid bilayer. On both sides of the plasma membrane, the inside environment and the outside environment of the cell, both are said to be hydrophilic. There is water. So the hydrophobic tails of the fatty acids, the phospholipids, are facing inwards in the opposite direction in the phospholipid bilayer, and the hydrophilic heads by the glycerol and the phosphate group, or facing to either side of the hydrophilic environment where the cell is exposed to. So hydrophilic is exposed to either inside or outside the cell, and this mid portion of the plasma membrane, the phospholipid bilayer, is the hydrophobic portion, where the fatty acid tails, hydrophobic ones, are facing in opposite directions. So Davson and Danieli suggested that all the membranes are composed of a phospholipid bilayer with this configuration of the two layers of phospholipid facing in the opposite direction, exposing the hydrophilic portion to either side of the environments, the inside and the outside of the cell, and in the midsection, the fatty acids, the hydrophobic tails, facing in opposite directions. Besides the phospholipid bilayer, Davson and Danieli suggested that there was a protein coat as an outer surface for the phospholipid bilayer. And this model was also known as the sandwich model, because if we take a look, Davson and Danieli model for the plasma membrane, it consists of a phospholipid bilayer, okay, with this configuration, and it has an outer coat made of proteins, this green layer on both sides. And we said that on both sides of the plasma membrane, the inner and the outer environment are mostly composed by water. That is a polar molecule. So this protein coat, it's made of amino acids, right? And when we talk about amino acids last semester, we said that amino acids can be either hydrophilic or hydrophobic. So if this outer coat of proteins is being exposed to both inner and the outer environment, composed mostly by water, this protein coat must be composed of hydrophilic amino acids. Okay, and that's the only way they could have been exposed and remain in that position. But if we assume that this protein coat is based on hydrophilic amino acids, then we must assume, guys, that all the plasma membranes for all the cells must be identical. So all the membranes have a phospholipid bilayer with a hydrophilic protein coat. Okay. Eventually, there was rejected the fact that all membranes are identical. No way. And since these proteins will be exposed again to these hydrophilic environments on all sides, inside and outside of the cell, this configuration was not stable for the plasma membrane. Again, the fact that the proteins are 100% based on a hydrophilic amino acids, and they are in the outer portion of the membrane, not possible, at least not for a stable structure of the plasma membrane.
And why is that? Why are proteins becoming unstable when faced with hydrophilic environments?
Again, we cannot assume that proteins are 100% made of hydrophilic amino acids. Okay? If so, okay. But the problem would be the fact that proteins, due to this uh, nature being 100% hydrophilic, they wouldn't allow some other molecules to move across the membrane. It wouldn't be functional. Okay, since they do not allow some other molecules to move across the phospholipid bilayer. I mean, even though the membrane is it has a protective function, guys. It has to be permeable. The structure of the plasma membrane must allow some other molecules to move across. And based on this structure and configuration of the protein code on both sides of the phospholipid bilayer, it wouldn't be as stable as the model that is currently accepted. So it was a pretty cool approach about the structure of the plasma membrane by Davson and Danieli. Eventually, it was rejected based on some evidence of some electron micrographs where they noticed that, like in this one, they noticed that based on the sandwich model, the interaction between the membranes of two cells wouldn't be possible, or at least not functional. Okay. So later on, there was another attempt to describe the structure of the plasma membrane, and this was by Singer and Nicholson. That is the currently accepted model for the structure of the plasma membrane. It also consists of a phospholipid bilayer, similar to the Davson and Danieli. Okay, in the same arrangement, the hydrophilic portion of the phospholipids exposed to either side of the hydrophilic environments in the cell, the inner and the outer environment, and both sections of both hydrophobic tails of the phospholipid bilayer are facing inwards in the opposite direction. Okay. So here's how the Singer-Nicolson model differs from the configuration proposed by Davson and Danieli. We can tell that in both models there's a phospholipid bilayer. That's what they have in common. For the Singer-Nicolson, that is known as the Fluid-Mosaic model, there are also proteins, but in a different configuration. Okay, let's see, guys. For the Fluid-Mosaic model by Singer and Nicholson, again, the phospholipid bilayer is the same. In the same configuration, same arrangement of phospholipids, hydrophilic heads on both sides, and in the midsection, the hydrophobic portion of the phospholipids, the fatty acids or the hydrophobic tails. And we can tell that there are proteins involved in the structure of the membrane, but they are not as a coat, as an outer coat layer. Instead, proteins are embedded within the structure of the phospholipid bilayer. Okay? This portion of the membrane, guys, the phospholipid bilayer, is it hydrophilic or hydrophobic?
Hydrophobic.
Hydrophobic. So you can tell that this portion of the proteins are in a yellowish color, right? This
So
yellowish
are they
part of the
also
protein.
hydrophobic?
Exactly.
I
This portion of the proteins is composed of hydrophobic amino acids. So they can interact with the hydrophobic tails. And another portion of the protein, this one that is either exposed to the outside or inside the cell, is composed of hydrophilic amino acids. Okay? So that we have a stable configuration of the phospholipid bilayer and proteins, okay, that make up the structure of the plasma membrane. On both sides, the hydrophilic portions of the phospholipids and proteins are exposed, and in this midsection is the hydrophobic portion of both molecules, proteins and the phospholipids. It's more stable. It's fluid. We said that the structure of the plasma membrane has a distinctive face. The inside, the inner face, and the outer face might be different, and this is due to this fluidity of the membrane, guys. Proteins are not static within the plasma membrane. They can move, they can float within the phospholipid bilayer. So it's dynamic and it's fluid. And some other times, proteins, and like in this example, guys, you can tell that protein, the whole protein structure um, is across the whole structure of the phospholipid bilayer. Okay, so one portion of the protein is on one side, then it moves into the phospholipid bilayer. The structure is different, so you can interact with this portion, and then another fraction of the protein is exposed to the outside. Some other times, some proteins are just exposed to one side of the membrane. They can expose just a fraction to the outside, but not to the inside. Or some other times, proteins can be exposed to the inner environment, but not to the outside. Okay? Either way, there is an interaction between the protein structure and the phospholipid bilayer, but in a different configuration. Are there any questions, guys?
Just to be sure, the Fluid-Mosaic model is more stable due to the fact that the two sides has this have this uh, uh, hydrophilic ends with a bit of hydrophobic proteins, no?
Yeah, just one fraction of the protein is hydrophobic.
Okay, now that's why the color
Just
changes.
exactly the yellow portion of the molecule of the protein, we assume that is hydrophobic, and the greenish part is hydrophilic. That way, when it's exposed to the outside, it has one composition, one structure, and then as it moves and fits in within the structure of the phospholipid bilayer, the structure of the protein, we assume that it's based on hydrophobic amino acids. And by this configuration, it's how the plasma membrane achieves this selective control of molecules. Based on their size, some molecules can freely move across the membrane through the phospholipid bilayer. Okay. Some molecules that are really large in size, they cannot freely move across the membrane. The cell requires another mechanism. Some other molecules, due to their chemical nature, in order to move across the membrane, they depend on the protein. There is a channel that is formed by proteins, and that's the only way these molecules can cross it, move across the membrane. So proteins play many roles regarding the function of the plasma membrane, and that's why the fluid mosaic model suggests a more suitable configuration for this function rather than the sandwich model.
Just another question. Why we couldn't assume with the sandwich model that all the cells had the same membrane, but why we can with the fluid mosaic model?
Because again, since proteins are the that's why it's called the fluid, and the fact that this
Ever changing.
model, yeah, the molecules within the structure of the phospholipid bilayer can flow. They can be moved from one place to another. So these proteins can move from this place to another place. They can remaining as part of the membrane, but they can switch they can change their place, their location. And it's a mosaic because we can tell that the composition of the membrane is not homogeneous. It is not all the same throughout the structure of the membrane. That's why the inner portion or the outer portion of the membrane can be different. Because we assume that different types of cells have different needs, different interactions, and as we increase the organization of these cells, the membrane plays a vital role regarding the formation of tissues and some are a more organized structure such as an organ. So we cannot assume that all membranes are the same because that's not true. It wouldn't be functional. And this model suggests that this structure and the composition of the membrane can change. So it can be adapted according to the type of cell and according to the needs of the cell.
Okay. Thank you. That was itching in my mind, but now I get it.
Okay. Okay, this is the way the fluid mosaic model looks like. Okay, or should look like. You can tell it's a complex. It has an heterogeneous composition. Besides the phospholipid bilayer and the proteins, there are some other molecules, guys. There are some other proteins that could be attached to a carbohydrate, or they might have a carbohydrate attached to their structure, and these are referred as glycoproteins. There might be some other lipids that might have a carbohydrate sugar attached to the structure. These are known as glycolipids. There are some other type of lipid besides the phospholipids and steroid, cholesterol, that plays an important role in the flexibility and strength of the plasma membrane. So besides the phospholipid bilayer and proteins, there are some other proteins, there are some other lipids, like glycoproteins, glycolipids, and cholesterol. So we can tell that, well, here's the phospholipid bilayer, cholesterol, glycoproteins, glycolipids. You can tell that the sugar that is attached to the structure of the lipid and the protein. Proteins that can be within the structure of the phospholipid bilayer. Some other proteins are just exposed to the inner environment. Some other proteins are exposed just to the outer environment. So we can tell it's an heterogeneous composition of the cell on either side of the membrane. And the activity for this week, guys,
Teacher,
is yeah.
can you repeat what glycoprotein was made of and glycolipid, please?
Okay, a glycoprotein is a protein that is a protein usually is made of amino acids, right? But a glycoprotein is a protein that has a sugar. It could be a monosaccharide or a disaccharide attached to the structure of the protein, like a complement. Okay. There are some
So
other
that's
lipids known
why
as
glycolipids
yeah.
are like attached to glycoproteins or not.
Uh, Glycolipids are lipids. Could be just could be a fatty acid, just attached to a sugar. Could be a phospholipid. Could be a triglyceride, maybe. We, there are different types of lipids, guys. But these lipids have sugar, a monosaccharide, disaccharide, okay, that is attached to the structure of the lipid. Okay, so they have different roles depending on the structure. And actually, most of these glycoproteins and glycolipids are used by the cell to be recognized by some other cells. That's why the surface of the plasma membrane for a given type of cell is some sort of, well, it has a unique configuration and structure. And that's the way cells can recognize as a part of a tissue, as a like cells that have a specific structure and function.